There are some things in biology that just make me realize how utterly nonsensical evolution is. One of those things that I recently learned about is alternative splicing. So that's what I'm going to be bringing as evidence against evolution today. And I've given many reasons that you should reject, many scientific reasons that you should reject evolution in previous episodes. None of those have been challenged thus far. And I welcome you. If you disagree with me, after I've presented my evidence against evolution, I invite you. You can challenge my evidence and you can also present your own evidence for evolution if you'd like. And if there's anybody else watching who wants to also give evidence against evolution, you're welcome to join. I'm only letting one person join at a time, so you may have to wait backstage if someone else has previously joined. Okay, so alternative splicing. What is this? Now, in the human body, for example, we have about 20 to 25,000 protein coding genes that we know about. But what's really interesting is that in the human body, there are many more types of protein machines than we have genes to code for them. So estimates, I could not find a good estimate on this. The lowest estimate I found is that we have 100,000 different types of protein machines in a human body. I found one estimate that was like 24 million. So somewhere in between 100,000 to in the millions, that's how many different protein machines exist in a human body. So how do we get all those when so far we've only found 20 to 25,000 protein coding genes? Well, the answer is very surprising, and it's called alternative splicing. And what that means is that, so let's just, up at the top, you see on this diagram, the colors that represent exons that are used to build a protein, okay? But what happens is that you have an army, hundreds of protein machines that mix and match those genes depending on what is needed. So whatever type of protein is needed, they take from what's already there and they mix and match them to make the gene the way that needs to be for the specific protein machine that's needed. Now, why is this a problem for evolution? Well, it's obvious to me, and it surprises me that people aren't automatically seeing this. So, because I know that many biologists just totally accept evolution as a fact. And so, but why is this alternative splicing a problem? Well, number one, it shows intentionality. It shows that there is somehow a know-how of what needs to be put for that cell and a specific intention, a goal to get that protein machine. And that requires planning. It requires knowledge, foresight of what is needed. And it requires these hundreds of proteins working together on the same goal. So where does this knowledge come from? And what's really interesting is when I watched various videos, read various papers on that, a lot of intentional language is used, even though evolution is not supposed to have any intention. There's not supposed to be a goal in mind. This is all supposed to be random, happening by random natural processes, mindless processes. But the words that are used for this process of alternative splicing all require a mind. So, for example, here's some language: recognition by the spliceome. Okay, recognition. Can a mindless process recognize? So, recognition is something that minds do. Facilitation. Okay, facilitate. In order to facilitate, you need to know what you're trying to get to. You need to have a goal. Help. Wait, Help for what? What is the goal here? There has to be a goal in mind. Attempt. Attempt is a word used. So that means that also implies a goal, that these proteins are working toward a goal. Mediate, provide, ensure, tolerate, select, drive. And so all of this intentional language is used to describe this process of alternative splicing. And so that actually is inescapable. And I want to show you a video that just kind of shows this process of splicing. I actually couldn't find one for alternative splicing. This is a video showing the proteins involved with the spliceosome bringing together the exons and to get rid of the introns that are not needed. And they are, as you can see, this process, it looks very intentional, looks like they know what they're doing, looks like they have a goal in mind. They're doing a very specific process. They're trying to get that green part, which is the green represents the intron that is not needed, and take out and prepare the exon for being built into a protein. And it's going to show another image of this in a minute. And then, so here you go. And it's showing it because there's more than one place on the mRNA where this is happening. It's happening here in 3 different places. And so you can see this is a very intentional process. So how do these hundreds of proteins know what needs to be done in order to get this transcribed to the correct protein? That is a big problem for natural processes to explain. But hey, we have evolutionary stories for this, right? Yeah. Well, a lot of people think that just because there's a paper written about the evolution of something, that is actually evidence for the evolution of something. When I look at these papers that are about the evolution of something, I usually find the best evidence against the evolution of that thing. And what I notice is that they always start with the assumption that evolution happened. There's no question here. It's just taken for granted. That's how deeply mythology has been embedded into what is called science today. And so there is no question about whether this evolved. It just is assumed to have evolved. And everything that is written is based on that assumption. Well, let's look at this paper called The Origin of Spliceosomal Introns and Alternative Splicing. And so this paper says, in this work, we will review the current knowledge on the prehistory, origins, and evolution of spliceosomal introns, and then going on about the alternative splicing. So how do they propose this happened? Let's look at what they say. They say in this section, we discuss, so by the way, I'm skipping the part where they try to do the origin of the introns because that's a huge section. And I wanted to focus on the spliceosome and alternative splicing. So the spliceosome is that big machine made of many proteins that is working on splicing. And then so they're going to talk about that, and then they're going to talk about alternative splicing. In this section, we discuss the major steps leading to the origin and establishment of the spliceosomal system in eukaryotes. Despite the persistence of disagreement on some important aspects, there is a general consensus about how this process may have unfolded. According to this general model, spliceosomal introns evolved from an invading group 2 introns, perhaps derived from the early mitochondrion. For some reason, these introns then proliferated to an unprecedented level in the host genome. Over time, the self-splicing activities of these many intron copies degenerated, which was associated with an increase of trans-encoded RNAs and proteins that promoted efficient intron splicing, setting the basis for of the protosplisomal machinery and further releasing selective pressure on cis-intronic splicing elements. As this protosomal machinery recruited more proteins and became more efficient, introns became increasingly reliant on the emerging spliceosome for proper splicing. Okay, here, this is all evolutionary storytelling. When I highlight something in pink, it's no science and all evolutionary storytelling. So they've laid out the evolutionary story here. They believe that the introns that are necessary in this whole process came from some other type of invading intron, and then for some reason these introns just kept making more and more and more in an unexplainable event, and just then they degraded, and then they're all of this was precursor for making a spliceosome and alternative splicing. So yeah, that's all just storytelling. Now here's more storytelling. And this is also regarding the origin of the spliceosome. Now, I just highlighted in green a couple of points that are important here. One is that these things, all of these things that are necessary for the spliceosome are present in all eukaryotic groups. Okay, and based on that, they say this had to evolved very early, that this was present in the last eukaryotic common ancestor. So at the beginning of eukaryotes, 2 billion years ago, all of this complex, incredible alternative splicing and spliceosome machinery all existed. It says by the time of the last eukaryotic common ancestor, a very complex, modern-looking spliceosome composed of at least 78 proteins and all the snRNAs had already been established. Okay, so this is another major problem. And that is, evolution is supposed to be this process where things are slowly evolving over time. But why are we always seeing the sudden appearance of forms? We see the sudden appearance of forms in the fossil record, and here we're seeing the way that the way that they're assessing whether this was there at the beginning or not is they're comparing genomes of living eukaryotes. And if some of them didn't have all this machinery, then maybe they would say, oh, this probably evolved later on. But because eukaryotes all have this amazing machinery, they say, okay, it had to be there at the earliest, the very beginning of eukaryogenesis. So they say it had to be there. So it had to be there like 2 billion years ago. And there's a sudden appearance of it because they say prokaryotes, evolved into eukaryotes or had a symbiogenesis, but there's no buildup, there's no transitional forms of this. This is a sudden appearance. That's a contradiction. It's a problem for evolution. So there's the sudden appearance of modern looking spliceosome. Okay, very complex modern looking spliceosome. So it looked the same as far as they can tell. It was the same. It's the same throughout all of eukaryotic life. It all looks like it was there from the beginning. Okay, so so Nessa said, oh, skip that section. Nice. Yes, I'm skipping a lot of sections because I don't think anybody would watch this video if I went through the entire paper. Okay, but you're welcome to come on and talk about any of the sections that I missed. If you think I'm missing some of the evidence for evolution, I welcome you, Nestle, to come on and share. And I will remind you that last time you came on, you did challenge the paper that I brought about the evidence against evolution of the ribosome, but you were not successful in showing me how this was evidence for evolution. But going on, intriguingly, the reasons for this transition from the intron retention to exon skipping during evolution of early animals is still unknown. Okay, so finally they're getting to the conclusions and they're admitting that they do not know how we got from intron retention to uh, exon skipping, which is, increasing the diversity through alternative splicing during evolution of early animal. Okay, so it's still unknown. Given the potential centrality of alternative splicing for the origin of animal complexity, understanding this crucial transition is a central priority for future studies. So they admit they don't know how this happened. And that's fine, but it's just important for you to know that there is no current scientific explanation for the evolution of the spliceosomal system and alternative splicing. Concluding remarks, evidence from biochemistry and comparative genomics strongly suggests that spliceosomal introns originated and proliferated during the origin of eukaryotes in a process intimately linked to other evolutionary events of eukaryogenesis. Again, they're just saying what they found before, which is, hey, it had to be toward the beginning. If like they since we see it in everything, basically that tells us it was part of eukaryogenesis, which is problematic because you got to have a lot of other things coming about at that same time, a lot of other complex machinery and differences coming about at that same time. The resulting portrait of the last eukaryotic common ancestor spliceosomal system was comparable to that of intron-rich modern eukaryotic organisms. In other words, it hasn't changed in 2 billion years, with myriad introns, complex splicing machinery, and coupling of splicing to other cellular processes. From that complex ancestor, intron and exon structures have been highly plastic throughout eukaryotic evolution, with cases of extreme divergence from their ancestors that shape the genomic landscapes of eukaryotes, probably like no other. Particularly remarkable, oh, I love this, are the multiple instances of recurrent evolution of multiple features, including massive intron loss, constraint of splicing signals, restriction of BP3S's distance, loss of polypyrimidine tract, sorry, my pronunciation, and complete loss of U12 introns. Okay, so I love that because, once again, like we see in all of our little looks at the phylogenetic tree, is that we have instances where features had to evolve the same way more than once. And so that is a problem. That's a contradiction. So now I have shared. Okay, now look at this. I love this. Mehrashad says, it seems to me there is no detailed evolutionary scenario for spliceosome machinery. It did surprise me. Yeah, but there's many more things that there's no detailed explanation for. In fact, you'll find that many of things that they are just assumed to be evolved without any evidence at all. Now, if anyone wants to join and share evidence for evolution, I welcome you. And I'm going to just remind you about the past evidence that I've shared against evolution that no one so far has challenged. Or not successfully challenged. I just shared alternative splicing, evolution of ribosomes, evolution of circulatory systems, or evolution of gene origination, DNA code, evolution of the DNA code, evolution of metabolic pathways, evolution of metamorphosis, coevolution of enzymes, evolution of helicase, nuclear pore complex evolution by Sal, collagen and blood vessels in dinosaurs, Cambrian explosion as evidence against evolution, millions of fish fossils, no predecessor to fish, thousands of dinosaur fossils, no predecessors to dinosaurs, 1,000 bat fossils, no bat predecessor, and many diseases caused by mutations, demonstrating that mutation is generally something bad. It's not a sufficient mechanism to make evolution happen. Now, here are the evidence that has been shared for evolution that I have accepted as evidence for evolution. And this is a new one that I added. Last week, Dr. Dan said that there's a mutation that doesn't cause sickle cell, but protects against malaria. So I accept that as evidence of evolution. I did not accept his presentation of lactase persistence as evidence of evolution. And the reason why is because when I looked into it, lactase persistence seems to happen very commonly in cultures that have that eat a lot of dairy. And so it's hard to say, was this something intentional that the gene regulatory network does in order to facilitate that? It seems like it is. And also because it's happened in like 5 different ways. So it seems more to me like an intentional change. If something is an intentional change, I'm not going to give credit to random mutation for making that positive change. Seems more likely to me that somehow the processes in the cell knew that this was needed and allowed for lactase persistence. So that's why I didn't accept that one. Previously, Erika made she discussed the prediction of ape-human transitional features, and we found those. I accepted that. Grayson presented the prediction of transitional reptile bird features, and then Archaeopteryx was found. I accepted that as evidence. Grayson said the prediction of transitional amphibian fish features, and we found Tiktaalik, which has a sort of a mosaic of those features. So I accepted that as a positive prediction and evidence for evolution. And that is basically all I wanted to say. If nobody joins, thank you, Wesley. Appreciate that. I'll just look back through some of the comments. And again, I welcome you. And I do want to remind you guys of the 4 scientific reasons. There are more scientific reasons to reject evolution, but the 4 scientific reasons that you should reject evolution are right here. And today we talked about number 3, integrated systems require foresight. And that is in alternative splicing, we definitely see foresight and we see intentionality. And I will just put that up on that video up on the screen again in case anybody is just hasn't seen this before. It's quite amazing. So, and I do have a religious agenda for doing this. I want to be honest about my religious agenda. My agenda is that I want you to know God. And unfortunately, I think a lot of people have been completely deceived by evolutionary storytelling that masquerades as science, and it blocks you from the main evidence that God has given everyone for his existence. When we look around at living organisms and at this orderly universe, it should automatically bring us to wonder and awe at the mind behind this. But unfortunately, some people have been taught these evolutionary stories and they've believed them so much that they can no longer see the evidence for God, and they wonder, where is the evidence for God? Okay, Nestlig, thank you for joining. How are you?
Oh, hello. How are you doing? I'm doing fine, but how are you doing?
Good, thank you.
I also wanted to thank you for taking my criticism about the ERVs in consideration. For the people in the live chat, like we also had a disagreement on the one episode you did on with Dr. Hunter. I had some criticisms on the piece of science forum, but you did take some criticism to heart and changed, amended a part of the video that you did with Dr. Hunter. So I want to applaud you for that.
Thank you. And I thank you for taking the time to do that. And I will point people to that article if you go on the Peaceful Science Forum. And I think what was the title called?
From the top of my head, I think it's Are ERVs evidence for evolution? Yes, exclamation mark. Yeah, it's pretty easy to find. It's probably like one of the most recent posts on the Peaceful Science Forum, so it shouldn't be too difficult to find it, even without a link. But yeah. Mm-hmm.
Yes, thank you. And so Nestlig critiqued me and Dr. Hunter's last presentation on ERVs, and we realized that we did make an error. And so I'm so happy that you did that, because I don't want to be giving any false information on this channel. So I really do appreciate correction on that, and we did edit it based on your correction. I know some of the arguments you made, I didn't really see a problem with what we said. I mean, obviously you have a difference of opinion.
We can discuss that. Take the time to discuss it point by point. Of course, I don't want to, like right now. I don't want to bulldoze you with all these points about ERVs. We can, like, more specifically, we can discuss the topic that you are discussing right now in this live stream, if that's okay.
Okay, yeah, whatever you want. I'm happy to discuss any topic that you want to discuss. Okay.
All right, so one of the things you said at the beginning about, like, scientists are often using like intentional language to describe the functions of, like, for example, these proteins. They recognize these sites where the spliceosome cut and paste the introns out of the messenger RNA, right? So, and I'm more like, well, you're taking these things a bit too literally. Like, for example, when we describe hydrophobic and hydrophilic interactions at the molecular level, we aren't saying that these molecules are like afraid or loving water, right? Like, even though we use the term hydrophobic and hydrophilic, we are actually saying that these molecules have a mind and that they are actually emotionally afraid or emotionally attracted to water, right? So it's a quirky thing of language where we have, like, these metaphorical meanings laid on top of one another. So that's one of the things I want to caution you against, taking things too literally regarding those things.
Right. And you're giving a good example of one that I wouldn't take too literally.
Yeah.
Okay. But what I've noticed is in every single paper that I've read about evolutionary processes, they use this language in stronger ways than, the example you gave, but ways that are inseparable from the idea of a goal. So, that is a problem, don't you think?
Like, in a way, like, I do myself have some problems with, like, for example, I'm a critic of the machine analogies often used to describe the world at a molecular scale. But yeah, so regarding those things, I can agree that sometimes these metaphorical language can be pretty misleading to most people. That's probably a good criticism of that as well. Yes.
Okay, so, but, like, for example, with this alternative splicing,
yeah, that's good. Yeah.
Can you give an example of how you would describe alternative splicing, why it happens and how it happens without using that type of language?
Oh, we can actually, like, I can pull up the paper that you showed
Okay.
a few minutes ago. Let's see here. Can I screen share?
Sure, of course.
All right. So it gives a. Let's see here. Hold on a minute. Screen share. All right. There we go. So yeah, it's a different. It's on the on PubMed this time, but it's just the same paper. So yeah,
Okay.
you can see this image right here. It's let's see here, just making it more eligible to most people in the chat. Let's see. All right. There's the image. You can see this image, right?
Yes.
What it depicts is, like, it's an example of a gene with introns denoted as, like, black lines, and the exons, the parts that code for protein, are given as these big black boxes. And what you see is a sequence logo. Basically, they denote how often a particular nucleotide are present in these locations, which is recognized by the spliceosome. Like, for example, maybe we can zoom in. Let's see here. We can zoom in. There we go. So, for example, it's not a very.
But do you see that you just said recognized? So can you use a word?
It's a short. Yeah, basically, what actually happens on the molecular scale is you have these physical interaction. Like, for example, one of the important interactions is the hydrophilic interaction that I previously mentioned. But here we're also dealing with like hydrogen bonds interactions. Like, for example, the specifically the spliceosome. Do they give an image on how the spliceosome works? Let's see here. Maybe they do give an example. Are you still seeing my screen?
Yes.
All right. Let's see here. Do they give an example on how the spliceosome actually works? No. All right. So I can just explain a bit. Like the spliceosome also has these short RNA sequences, and these short RNA sequences, when they find a complementary sequence on the messenger RNA, I will go back to the image. When they find a complementary sequence, for example, right here, they will recognize this, and if these sequences are compatible with the messenger RNA, then that is when the spliceosome is able to cut the RNA at this specific location. So this is and that involves hydrogen bonds.
Right, but
Yeah.
did you notice? And like, since I wouldn't pick on you too much about this, except that this was the thing that you brought up about the language. And I'm saying we are using intentional language, but trying to make it non-intentional. But what I would like to show you is that you can't really take out the intentional language. You said find.
I can do it.
Mm-hmm.
I can do it, but of course it will take much longer to explain than just use recognition as a shorthand for the whole process. Like we are dealing with hydrogen bonding between this messenger RNA and a short RNA present in the spliceosome. So, but we can just abbreviate this as a recognition point. But I'm not sure what the different words we can use. We can. What else can we use? Yeah, it's probably also a bit of a quirky thing with language. Again, we're also using these terms with hydrophobic and hydrophilic interaction as well. So it's probably we are just stuck with these words that we are accustomed to using, and sometimes these metaphorical implications can get omitted sometimes. So. Mm-hmm.
Yeah, and Mershad makes an interesting point. He says, but molecular interaction needs to be regulated, which ultimately leads you to design language. But this is the exact point. What's doing the regulating? In order to regulate, there has to be someone who set the system up. There has to be, there has to be a regulation system. There's no natural process, mindless process that cares about regulating. So yes,
actually can be, because there are many systems that have, or many natural systems that have natural feedback, which regulates the parameters of the system itself. So it's self-regulating in that sense.
Okay, isn't that interesting that these introns and like little, hydrogen or whatever you're saying that is being recognized, okay, is
basically like you are familiar with the base pairing, like a G pairs up with a C and a T pairs up with an A. And the reason why this is the case is because the hydrogen bonding the hydrogen bonding between G and C is more compatible than, for example, with G and T. It can happen. So sometimes you have like a weaker interaction between G and T, or G with A, but G with C is a much stronger interaction. There are more hydrogen bonding available when you pair up a G with a C, and that's why. And then the same is true for this T. The same is true for this A, and the same is true for this T. And you can also notice in this I don't know if you're familiar with the sequence logo. Basically, it shows that this depicts how often we find a G at this location. Like in this location, we find a G pretty much all of the time. This G is very important at this location, but we can see here, like, oh, this T And maybe I can zoom in a bit. Let's see, can I zoom in?
I get the idea.
Yeah. So we can see here, this T is also important, but sometimes we find a C at this location. So what this means is that this location is less important, but still of course important, because we do find a heavy bias towards the T location itself. So there is some leeway regarding the recognition. Like sometimes it can deal with a C sometimes, but it will be weaker. And the whole thing that I want to explain is like basically the system recognizes based on the strength of the hydrogen bonding that occurs between the messenger RNA and the short RNA present in the spliceosome. And we can just abbreviate that by, it's recognizing, but in the physical world, it's basically just the strength of the hydrogen bonding that happens between these nucleotides.
Okay, so then, like, if this is just all just a natural chemical process, how does it end up that these natural bonds end up forming exactly what the cell needs, the exact type of protein that the cell needs?
Well, it's like specifically what you mean by exactly what the cell needs. Like we have like one example, like sometimes, for example, we see here a one example where there is some leeway in the system. Like this T is less important. Also, others are still important because it's 1 bit at least, but still it's less important compared to the first position where the G is pretty much always there and always needed. So it's also like there's also one point that I also want to mention is like the spliceosome is not perfect. There are errors. Sometimes the spliceosome does not cut a messenger RNA at this location, and that results in these splice variants, which do occur sometimes.
Right, but what's amazing too is that we even have a little protein that engulfs the proteins that are not created properly and then destroys them. If it can't fix them, it destroys them.
Right.
So another amazing feat for natural processes.
Right.
Now, Otangelo wanted to ask, he said, what came first, the splicing code or the spliceosome?
I'm not sure what he's referring to, the splicing code. Maybe he's referring to this sequence.
Yeah.
I think if I remember correctly, as you mentioned in the before, the article this article that we are showing right here, it discusses the origin of the spliceosome from self-splicing introns. Let's see here. If you go, what's the part that you mentioned before? Can I? Yes, here. There's a group. I think this is the part that you mentioned, or maybe it's a different section that you showed before. Like, what was the section that you showed before?
I tried to skip the introns because that was a whole section. I was trying to focus on the spliceosome and the alternative splicing.
Let's see here. What was the? I think this is the section that you showed before. Like they discussed that these. Let's see here. It is a good section. Like they discuss that the origin of this spliceosome comes from group 2 self-splicing introns, and basically these introns, as the name suggests, they are self-splicing. They don't need a spliceosome to splice themselves out, and they are found in the bacteria in this case. And I think they also mentioned later on that the like in this section, just reading it out loud, the structural and functional evidence suggests that the spliceosomal and group 2 self-splicing introns are evolutionarily related. Both types of introns are spliced through a similar two-step catalytic reaction that relies on an endogenous adenosine, or PP, and releases the excised intron as a lariat structure. So basically, yeah, it's a very similar mechanism for how these introns are spliced out, and one does it, and one does it without the spliceosome. So I think to answer Otengelo's question, the quote unquote code preceded the spliceosome itself.
Well, now what's interesting is throughout the paper, it talks about how all of these features are present in eukaryotic organisms. And so, I mean, there maybe are a few exceptions where it has been lost or something like that. But in general, they think all of this had to be in place at the last eukaryotic common ancestor. And I didn't put up the quotes regarding introns on that, but that is what they said about the introns. So all of this seemed to have suddenly appeared. And what you read was there anything in that that actually supplied evidence to you of evolution? To me, that looks like it describes the process that they're using. It doesn't describe how that process could have originated. Are you talking? Because you're muted.
Oh, sorry, I was muted. I
Okay.
sorry. I want to mention that like in this specific section that I just highlighted is that they do discuss how similar the cell splicing or cell splicing introns are to the spliceosome operation. So there is some evidence at least to how they of course, like it's also one of my criticism that I have. Like you previously you showed, I think you highlighted this paragraph.
Yeah.
And then you skipped this and went straight on to another paragraph. And here is some of the discussion that they have regarding why they hypothesize that the group 2 self-splicing introns.
Well, like I said, if I skipped something that actually provides evidence, let me know. But so far, I didn't see anything in here that actually provides evidence. If I saw something that actually provided evidence, I would include it.
Well, in the next sentence, it's the next sentence right here that I stopped right before the sentence. But in this sentence, it says the two intron types, like the cell-splicing introns and the introns in eukaryotes, the two types have similar boundary sequences. The GT-AY in group 2 introns and usually the GT-AG in the spliceosomal introns, although some U12 introns are AT-AC. So there are some similarities regarding the code itself that Otengelo probably was referring to. So, but you can say that this is not sufficient evidence to conclude this, but then you just skipped over it, and I would really recommend you just discussing, like go over the evidence that they discuss, and then you discuss the evidence on your show instead of skipping over it. That would be much better.
Right. Well, Nest, like I said, I get out the highlights of the paper because I don't think most people will sit and read the entire paper. But I welcome people to do that. And I'm doing it with you right now. But I wanted to say, like I said, if I recognize this as evidence, I would have put it. But I don't see similarities as evidence for ancestry.
Even if you don't recognize it as evidence, you should still like show it and then discuss why you disagree with why this is not evidence for their conclusion, instead of skipping over it. And you said that, if I discuss these details, then most people will not watch it. Well, I find it a very bad reason to skip this important part entirely. So I still highly recommend you just look at what they show to support, even if you disagree. Just look at the parts where they give support for the hypothesis, and then show it to your audience, and then you can explain why you still disagree with why this is not evidence for their conclusion. But right now, or before I joined, you skipped over that part. And if I did not show up to your live stream, you would still not have discussed this part. So I still want to recommend you to discuss the evidence that is shown in the paper on your own. Even if I'm not here, you will still learn some of these parts that they are included in the paper.
Well, thank you for your recommendation. And I'm glad you're here to point out what you see as evidence. So, can you explain to me how the similarity is evidence of the evolution of this incredibly complex machinery?
Well, like this is also related to Otenjo's question. Like he asked a question about which came first, the sequences in the introns that are recognized by the spliceosome, or the spliceosome itself. And here we have an answer to that very question. We have actually an instance of self-splicing introns that use similar sequences to cut themselves out, but they don't rely on the spliceosome itself to cut themselves out. So that is a very good basis for a hypothesis where we can say perhaps the self-splicing introns are the precursor to the spliceosomal introns that we see in eukaryotes. Of course, that's just the beginning. There is a lot more research to be done to explain exactly how the spliceosome evolved, because the spliceosome also involves proteins as well, and that's a whole different story to how those proteins came about as well. But we have at least answered one question that Otenjo asked: which came first, the sequence motifs, like this one or this one, or the spliceosome itself? Well, it says that these group 2 cell-splicing introns do have similar sequences, but they don't rely on an external protein to cut itself out. It's self-splicing, as the name suggests. Yeah.
Yeah. Do you have a problem, Nestlig, with the papers, all of these papers, assuming the evolution of these structures before we have evidence of the evolution of these structures?
What do you mean they assume evolution?
Well, I mean, the title of the paper is Origin of Spliceosomal Introns and Alternative Splicing. And then it says we're going to explain the evolution of all this stuff. So it assumes evolution.
I'm not sure why the title itself says they assume evolution. If they give just an assumption without any evidence to back it up, then I would agree. But I don't see how the title by itself shows that they just assumes a conclusion. Again, in the abstract, just to review in the abstract, in this work we review the current knowledge on the prehistory, origins, and evolution of the spliceosomal introns. First, we briefly outline the major features of the type of introns. All right, so good. And also emphasizing the non-spliceosomal self-splicing group 2 introns that we also just discussed here before. Next, we discussed the main scenarios proposed for the origin and proliferation of the spliceosomal introns, and an event intimately linked with the origin of eukaryotes. We then summarize the evidence that suggests that the last common ancestor of eukaryotes had remarkably high intron densities and many associated characteristics resembling the intron-rich genome. So far from what I'm able to get from the abstract by itself is first they summarize how spliceosomal introns and cell-splicing introns are working. Then they say we discuss the main scenarios proposed. So they don't assume these scenarios. They discuss these scenarios and discuss what are the evidences proposed for these scenarios. So to me, they aren't by fiat saying these scenarios are correct. They are discussing it and discussing the evidence proposed for them. So I'm not seeing a problem with how they are worded here.
Okay, what about
Maybe your complaint is that they don't discuss any design scenarios for the spliceosome. Maybe that's your problem, that they don't discuss any design scenarios for the spliceosome.
Well, there's no other possibility offered. So it says intimately linked to eukaryogenesis. So basically, the assumption is that we have all of this complex machinery that it looks like, if the evolutionary story is correct, it was around 2 billion years ago, and it suddenly appeared. So why isn't there a possibility of an alternative scenario? Like, hey, we really can't explain how this machinery could come about by a natural process. It's possible there's another explanation.
Well, people are still welcome to propose an alternative. Although the problem is that when we discuss like intelligent design or creationism, and we ask like, what is the design scenario? Like, what was the process of manufacturing the spliceosome by a designer or by a divine deity? And I have not seen, like in this paper, they propose like one scenario is the spliceosome is the evolutionary precursor to the self-splicing introns found in bacteria are a proposed precursor to the spliceosome. But then when we ask, okay, like what does intelligent design or creationism bring to the table? And they don't give a scenario at all. They just say, oh, well, it's designed. And okay, like how was it designed? Like what was the process of designing? What was the process of manufacturing these spliceosomes? These questions are not even asked among the creationism or design community. So you can argue that the evolutionary scenario for the spliceosome is still incomplete. Like I would agree, like pretty much most of science is incomplete, but still we can say that the evolutionary explanation for the spliceosome is still incomplete. But design doesn't even have a proposed scenario at all, which we can test or discuss. Like, do you have any design scenarios that you are aware of for the spliceosome?
And I agree with that complaint. So, I would say there is a limit and to what we can learn about that. But at the same time, I think that we can look at the features of life and say that with confidence that it shows it is the product of an intelligent mind, that it is not the product. I think we can actually, at this point, rule out natural processes unless we discover some natural process unknown to us at this time. We can rule out the natural processes that have been proposed as
that's the problem with that argument. To me is that like if you say that the strength of a design hypothesis comes from the absence of a natural explanation, then that is a negative argument, and that doesn't really work to me.
No, I'm not just saying that. I'm saying the positive evidence. So like, yes, we can rule out the natural processes that we know about, but we can also look at properties of intelligent minds and what they produce. And we can say, okay, life looks like the type of product that comes from an intelligent mind. And I think that's what even all of these uses of the words, the intentional words, the recognition, facilitation, help, attempt, all the words that I mentioned, they are actually demonstrating our awareness that there's intelligence involved in this.
Like I already, like we already discussed, like the terms hydrophobic and hydrophilic, and I don't take those words to imply that a water molecule has a mind. That's
And
the
I agreed with you on that, but I
yeah,
asked
yeah,
you to
right, exactly.
provide, can you find a word instead of recognition and find that you were using, and you
Well,
had a hard
the
time
problem is
doing
like
that.
again, I think the problem comes down to like the very vocabulary that we are stuck with the current vocabulary that we are having accustomed to. Like in molecular dynamics, we use like again hydrophilic, like for example, when a magnet, a positively sided magnet attracts a negatively sided magnet, we use the word attract, which again, we are not saying that they are in love with each other. we are using these metaphorical meaning to describe what happens, and it's just a quirk of language to me that we are using these things in a metaphorical way, and we should be very careful to not imply there is to imply anything else from just describing what happens, instead of implying that there is some that a magnet has a mind in of itself, of course.
Right. Well, that would be another example of maybe we have different uses of the word attraction, right?
Yeah,
So that would be an example of one that it could be used in the way we use it between people, but it also has a meaning that can just be like a chemical attraction, or
right.
you know.
And the same goes for like I think we would argue for same goes for recognition. Like we can describe like, oh, I recognize you as Rebecca, but when a sequence of DNA matches a complementary sequence of DNA, and they are able to form a double helix, for example. We can describe it as, oh, they are attracted to each other, or they are recognizing each other. But of course, we are not giving the same mental or the same mind properties to these pieces of DNA or RNA. Yeah.
Right, but the result of it looks like something that's the product of a mind. So like, by what process do you think these genes are being selected in a way that will be beneficial to that particular cell? So like, if we just take the normal process of, okay, this gene codes for this protein, right? But in alternative splicing, we have an intentional rearrangement of those and
Well,
leaving out of certain things. It's like a chef choosing certain ingredients for a recipe, or me choosing clothes out of my closet.
Well, I would disagree with the description, because like when I look at the evidence for alternative splicing methods, like I would actually say that most instances of alternative splicing is actually errors in splicing. And as I mentioned before, the spliceosome does, quote unquote, recognize these motifs that I previously showed, these sequences in the introns, and the spliceosome recognizes those sequences, but sometimes it fails to cut those places, and then you end up with these alternative transcripts. And most of the time, there are some exceptions, but most of the time those transcripts are non-functional, and they are just digested by enzymes that will digest the messenger RNA that result from the errors in splicing.
Yeah, and if this was all non-intentional, non-planned, wouldn't we expect most combinations to be non-functional? Like the vast majority?
And the vast majority of alternative transcripts don't produce a protein.
Right. So that's what I'm saying. So isn't it kind of interesting that we have so many different kinds of proteins, somewhere between 100,000 and millions different proteins operating that our body needs?
Um, Are you saying that that most alternative transcripts to produce a functional protein, or like maybe I'm wrong?
No, I'm saying that somehow we're getting the selections are being made properly. So how is it that we end up with what we need?
I'm not sure what you I think are we discussing protein production in general, or are we discussing alternative transcripts?
Well, I'm discussing the alternatives, but okay, so I just want to put up this comment before it gets lost.
Oh yeah. All right.
Wesley said all models have to be assumed in order to operate. So I think he's helping you out there. I mean, he's siding with you on that. So thank you, Wesley. I want to talk to you more about that. Okay. So, okay, we're talking about the alternatives because I'm saying, look, we have all of these functional proteins in our bodies, 100,000 to millions of different functional proteins. Most of them did not come from a straight up reading of the protein coding gene. It came from rearranging.
Oh, so I see. Like I think we are talking past each other. Like we have about 25. Like if my memory is correctly, we have about 20,000 to 25,000 genes. But you say we have like a million proteins, and to get those million from those 25,000 genes, we need alternative transcripts to get those. But
yes.
I think my memory is not fresh on that, but if I remember correctly, I think you may be confusing the combinations of the. For example, you have 3 separate genes, and the proteins coming from those 3 separate genes, they can form different protein complexes in different combinations. Maybe you are talking about those types of No, all right.
No. All right. I'm talking because, I mean, there is an example of that we just talked about, which is the spliceosome, which is made up of many proteins that are put together. But what I'm talking about is, like you mentioned, that we know of, we only have between 20,000 and 25,000 protein coding genes that we know about. But in our bodies, we have at least 100,000 and probably many more different types of proteins, which tells you that most of those proteins are coming about by alternative splicing. So that's the mixing and matching of those, protein coding genes. And so I'm saying that shows intentionality. There's an intentional picking out and splicing together the gene that will make the particular protein.
Oh, so I think I begin to understand. You still acknowledge that most alternative transcripts don't produce a functional protein, but still we need those alternative transcripts to explain why we have so many more proteins compared to the number of genes. Am I?
Well, there's one point we need to tease out a little bit. Are you saying most of the alternative, like the protein formations, like that are possible are non-functional, or that are chosen are non-functional?
Just to name a one, like if you have one gene and which is transcribed into a messenger RNA, what the researchers do is like they take the RNA, they do reverse transcription to DNA, and then they sequence the reverse transcribed DNA, and then they see these transcripts from the messenger RNA, and sometimes they can see like, oh, we mostly see this version of the transcript where most of the introns are there, or most of the introns are spliced out, and most of the exons are remaining. But sometimes we see a version of this messenger RNA where 1 or 2 of the exons are also spliced out. And I think there's also the image that you showed in the beginning where you see these alternative transcripts where some of the exons are also spliced out. But most of the time when that occurs, where an exon is spliced out, that is an error most of the time. But I can Like I can perhaps bring up a paper that I just found maybe an interesting paper to that you can also read yourself. Let's see here, how many human.
Yeah, I'm just trying to figure out because it sounds different than everything that I've been looking at, and I haven't been, and I'm not saying that there's not mistakes that are made, but
Right.
but it sounds like that in general, this alternative splicing. Hold on, I have to. Okay, the alternative splicing is specifically mixing and matching for proteins that are needed, and leaving out exons for intentionally.
Sorry, I was muted. Okay, you see my screen right there?
Yeah.
Like I just found this paper, although unfortunately it is it's paywalled, but it seems to very relevant to discussion here. They discuss how many proteoforms are there.
Yes.
Here it asks, we use this framework to assess existing data and to ask the question, how many distinct primary structures of proteins or proteoforms are created from the 2,300 human genes. We also explore the prospect of improving measurements to better regularize protein level biology and efficiently associate PTMs to function and phenotype. All right, so it's although right now it's like I need to open the paper up to fully explain this, but I don't think we have time to go into this paper. Maybe it's a good idea to like put this subject on hold, and then we can perhaps discuss this subject in more detail if you want on the Peaceful Science forum. Maybe you can like I won't write a whole a whole thread on Peaceful Science. Maybe we can do a separate thread on Peaceful Science to discuss this topic in more detail.
Well, how about if we do it on my channel? Because I already went off of the Peaceful Science forum. I asked them to delete me.
Oh, why?
Because it seemed like there was a lot of very angry people on there. And I just don't have the energy to go back and forth with them in written comments. Okay.
All right. So if that's the case, I like again, if you don't like to interact with those, just ignore those people. Just say like I only want to focus on this person, but I won't respond to anybody else. You can say that if you want, but of course it's entirely up to you. If you don't want to be on the forum, that's entirely up to you. Yes.
Yeah, well, I just I mean, if you want to come back on and talk about it, I would love to. And if you write something on there, I can go and read it. I may not respond on there.
But because right now, I tend to be like I tend to be more meticulous about the details, and if I'm not sure about, like, for example, it seems to be a very interesting paper from Nature, where they discuss this very subject, but I don't know exactly what they have done in the paper to this. So perhaps we can discuss this in more detail. It will take more time than we have right now, of course, because right now we can maybe read the whole paper, but I think you maybe want to move on to a different person if there's a different person.
There's nobody waiting. There was somebody waiting backstage earlier, but there's nobody back there right now. So if you leave, I'm just going to shut it down. Or if anybody wants to join, now is the time. And and Wesley, I would love to hear from you. Now, I'm not sure if Wesley's asking this. I mean, to me or to you. So you agree all of science assumes a model to operate in.
Like I think the point, like if I want to steel man his point, I think he's saying that like an individual scientist cannot like start from scratch. They have to like stand on the shoulders of giants, as we often put it, in order to progress science forward. Like when a physicist does an experiment, they don't go all the way back to prove the existence of gravity, for instance, right? They like that work has already been done before, and they just move on to the next point of contention. So that maybe that is what he means by it. Like they take some things for granted, and then they build on top of that. basis. Of course, we don't say that gravity is gospel. We don't say that. We are still open to any corrections on those basic established principles. This is
So
always
if
open.
we don't say that about gravity, why do we say that about evolution? Why are there so many things saying
I don't think we
evolution is a fact? Evolution is a fact. If you don't believe in evolution, you're not scientific. Why do we do that?
Like for the same, like we can also say for the same thing with gravity. Like we can say that gravity is a fact because it's very strongly actually in a very ironic way. Evolution is more supported than gravity because
wait a minute, Nestlig, if evolution is that well supported, why can't I find the evidence for it? Why is it that I've done 16 of these shows, every time presenting strong evidence against evolution that has not been challenged, and then and you might be on the way to challenging what I'm saying, but you haven't actually challenged my core problems with this yet. So my evidence hasn't been challenged.
It's actually a recurring problem because just like last time when I came on, you discussed a paper showing an evolutionary scenario. You show some highlights about, this is just evolutionary storytelling. This is evolutionary storytelling. But then I noticed you don't discuss any of the basic points that they show to support the model that they are discussing in the paper. So you jump over you see a paper, you jump immediately to the conclusion, and you say I disagree with the conclusion of the paper, but then you don't discuss the evidence that they are showing in the paper, and that's the very problem that I have with how you are dealing with these papers.
Okay, well, next time I will try to look harder for something that you might consider evidence and post that. But
no,
again,
not me. Just discuss what the authors of the paper discuss as evidence. Even if you disagree that it's not evidence, you still for example, say this, according to the authors of this paper, this is the evidence that they provide. But here is why I disagree. That would be much better than what you are doing now. Okay. And what you're doing now is skipping right over those points. That's the problem I have.
I take the feedback. I'll work on that. And so Wesley says that's in response to what you said before. He said that's true, but that's not what I'm saying. Wesley, I would love if you would come on and talk about this if you can. He said, what I'm saying is that you don't look at any particular evidence aside from an interpretive framework. And yes, Nestlig, now that's my feedback to you as well, is that I feel like you're hearing what I'm saying, but you're like, have you heard that? Basically you're ignoring the strong evidence against evolution, and you're embracing these little things that are basically nothing burgers that are offered as evidence. So are you able to think outside of the framework and say, last time we had the discussion about ribosomes, and to me it's to assume a ribosome could evolve by a natural process, or to assume alternative splicing comes about by a natural process, it's a huge assumption.
Well, it's one could argue that it's a basic assumption in science because this goes into how do you define a natural explanation or a natural scenario. We in science, we can only test natural scenarios in a way, because if you propose a supernatural scenario, then how do we test those things? If the way just to give an example, if God created the ribosome by just boom, there it is. We cannot test that scenario in a scientific manner. Yeah,
that's true.
It's so in order for science to function, we have to assume that the explanation we are looking for is testable. Otherwise, we are stuck in a way.
But what if the truth is that God did create the ribosome? How would you figure that out?
And that's actually a job for the creationists to answer. Like they are proposing that scenario, so they need to propose a way to test that hypothesis. It's exactly what I was asking a few moments ago with you. Like they need to make a positive argument for design and not a negative argument against evolution. That's the problem I have with design creationism.
What if it was a divine, non-testable event?
Well, then we are stuck. Then we are
Yeah, we're stuck.
that's yeah, we are
But
stuck.
would
We
you
cannot move forward.
let me ask you this? What would you rather have? Your science or the truth?
Well, then you of course you assume that we are able to tell the difference. Like, how would you able to tell the difference? Like, oh, right, oh, we cannot move forward because it's a miracle. We cannot move forward. Like, but how are you able to tell if it's a miracle and we cannot move forward anymore? Like how? Like,
Well,
what's
I
the
mean,
point?
there's nothing wrong with continuing to move forward to try to explain things.
Yeah.
But like, I don't think if we cut off the explanation, like if you cut off potential explanations because you can't test them, then you're endangering yourself of not being able to find the truth. You've limited the amount of you've
Well,
limited the type of explanations that you can.
the reason why is because, like, if you're not able to like to test them, or maybe or even in the philosophical sense, you cannot be able to like tell the difference between an unexplained phenomena like you cannot tell the difference between an unexplained phenomena or a phenomena that cannot be explained. And do you understand what I'm trying to say? Like, if you cannot tell the difference between those two, then you are like then saying that the phenomena that is unexplainable, you cannot say that is the truth. Like, how would you be like at what point can you tell, like, oh, the truth is that we cannot explain this anymore? It stops here.
Well, I mean, I would say that that is kind of like we test the limits with the scientific method, but eventually when
But then
something has been
oh, sorry.
tested many times, if when something has been tested many times and comes up with a certain result, then we end up saying, okay, this is a law, right? So like right now we have the law of biogenesis because we've tested so many times that and discovered life does not come from non-living things. And so right now it's safe to assume life does not come from non-living things. So we have the law of biogenesis. If something ever violates that law, then we can change our law. But right now, life does not come about by non-living things. So does that make sense?
Yeah, but this goes back to what I mentioned in the beginning of a few moments ago about the negative argument. Where right now you're using the negative argument to argue for a supernatural scenario. Like, oh, we have tried natural scenarios so many times, it has bared no fruit of success. So at this point, I'm just going to call it and say it's supernatural, and then we give up. And to me, I don't I think that is a mindset that will stop science in its tracks. That's my opinion on the matter.
Okay. Do you believe in the law of biogenesis? Do you accept
Uh,
that?
According like the law of biogenesis was formulated by Louis Pasteur, right? So that was the proposed like what he said. Like, he was specifically testing the hypothesis that like decaying matter could have turned into new life force. Like, for example, if you have a piece of rotting meat, then the life force remaining in the rotting meat will transform non-living matter into flies, for example, or maggots, for example.
Yeah.
And that's like one of the hypotheses he was trying to explain. It is not like the spontaneous generation scenario is not life from non-life. It's actually relying on it says it postulates a life force emanating from decaying meat, for example. And Louis Pasteur was saying like, no, maggots do not come from rotting meat. They come from the previous generation of flies who are infecting the decaying meat. So that. So
Okay, let me.
it's not related to the origin of life in specifically. And in fact,
Oh, I
Louis
think it
Pasteur,
is. Do you know any violations of that? Like, do you know of any scientific evidence that shows life can come about by chemical processes? Is there anything that demonstrates life comes about by
so
chemicals,
basically
non-living
the question
chemicals?
like we have not solved the origin of life. That's yeah.
Right.
I
Okay.
would
So right now,
I
the
do.
scientific view, right now, if you want
Yeah.
to be scientific,
Yeah,
you
we
accept
cannot
the
right now
idea that
we cannot
yeah.
explain like that. Although I do like I do have some hypothesis I do favor regarding the origin of life, but of course I will still say in general we have not solved the origin of life as of now. Yeah, but then
Right. So is there any level that you could
but
come?
then the question becomes how do we move from here? Do we say we give up, or do we say like we keep going?
Well,
What
you
do we
can
do?
keep going if you want to, but would there be like would there come a point when you would say, "Okay, I've tried, I've spent many years trying to find a way to get chemicals to come to life. I can't find it, and therefore I'm going to have a different conclusion about life."
Specifically, a different conclusion that is not
That it
testable.
was
You mean?
created. Yes.
Yeah, that I don't. Again, I find that a mindset that traps yourself into a corner, and where you can no longer improve. You can no longer you can no longer move forward.
Well,
At
but
least to
see,
me.
or you
Yeah.
could see it this way that maybe this mindset that everything has to be testable is blocking you in a corner. It's keeping you from seeing the truth about life. Like, for example, okay,
Like
let me just give this
uh,
scenario. I want to give a scenario. I want to give a scenario and see what you say. Okay, I agree that lightning in general comes about by a natural occurrence of, various physical processes, right? But if there was like a random person on earth who said, "God, if you're there, strike me with lightning right now." And at that moment, not a cloud in the sky, he got struck with lightning. What would you conclude about that?
um,
Would you be open to the idea that that was a divine event?
well, in a way, no, actually.
Okay.
Because you like first you need to of course calculate like what are the odds of a person being struck by lightning, and so
Okay.
sometimes actually sometimes lightning does strike a person even though there's no clouds above it because it's red. But in any in any case, it's also like if you say that occurrence happens again and again and again, like every time a person says strike me with lightning, God, then that would be something. Yeah.
Well, that's where I was
We can
going next.
then
How many times would that have
that's
to be
something we can
repeatable?
test. That's something it's actually a very good question. Like what if we have a system, like just to bring up another example, what if we have a system of magic that actually works? Like if we say abracadabra and then a plate of food appears on your table, that would be testable. Then in that sense, magic becomes real and testable in that scenario. So yeah, so that actually comes around to what I'm saying. If it's testable, then we can move forward at that point. If it becomes testable, yeah.
Right. Okay. But if the lightning thing only happened once and you were trying to figure out why did this happen and you exhausted all your natural explanation, you know,
Then we cannot. If it only happened once, then we in the entire history of humanity, then we can, and it never happens again in the future. Then we cannot say anything. It's one-off example, and we cannot really say anything with statistical confidence about it.
Yeah.
Yeah.
What if that's the truth about how we got here? That it was a one-off event.
Well, in a way, that's true. Yeah, that's true for everybody who lives, right? Because like, if what are the odds of your parents meeting and having you as a child, and how many potential siblings could have been born in your place? The chances are astronomical regarding how unlikely it is that you were born as well. Yeah. So the hypothesis is that your existence must have been destiny. That's not testable in that sense. So yeah.
Yeah, and that one's kind of weird because there's so many possibilities, right? Like, but like when we're talking
Like,
about like life forming itself into these complex systems by natural
oh,
processes, to me, it's obvious that
there's
that
like
that came about by
just
an
to
intelligence.
just to like as a off the cuff statement, like you don't have to buy anything that I'm about to say about the origin
Okay.
of life. Like the one of the like a few of the scenarios that I favor actually proposed that the origin of life was not an accident. It's actually a how do you call it? It is a it's like a ball rolling down a hill, because the way life forms is it's a way to relieve an energetic stress on the environment. It's like your example of a lightning bolt. Like, why does a lightning bolt happen? Like do you know what a lightning bolt is in its constitution? Like it's a beam of
Electricity.
plasma.
Mhm.
Yeah, I also electricity. Like the ambient air around you is not conductive to electricity, but the plasma channel in a lightning is conductive to electricity. And the reason why a lightning bolt comes into existence is because, like, when you have a thundercloud, thundercloud separates charges. So some parts of the cloud becomes negatively charged, and some parts become negatively charged, and that is an energetic stress that builds up, and eventually the atmosphere can no longer hold the stress apart, so it breaks into a lightning bolt. And the same equivalent thing has been proposed about the origin of life. It's a chemically equivalent of a lightning bolt, where a stress in the environment can no longer hold itself back, and then life comes into existence to relieve that stress into, yeah. Yeah. And so and this specifically the stress I'm referring to is the like, I don't know if you're familiar with redox chemistry. Are you familiar with redox chemistry in any
No.
sense? All right. So one aspect of chemistry is reduction and oxidation. In simpler terms, it's just the moving of electrons. Like, you have a compound that donates electrons and one compound that accepts electrons. And the way that in our bodies, that it's basically food. You eat food, carbohydrates, and those are the electron donors. And the electrons are donated to an electron acceptor, which is oxygen. And that's the compound you breathe in. So that is the reason why you eat and breathe, to facilitate the redox chemistry that goes on into your own cells. And an Earth-like planet without life has this energetic stress regarding redox chemistry. So the interior of the planet is rich in electron donors. Iron is a good example of an electron donor inside the Earth. But on the outside of the Earth, in the atmosphere and the ocean, what happens is solar radiation interacts with the upper atmosphere, and what happens is it will break apart water molecules into hydrogen and oxygen. And hydrogen is a very light element, so it will escape into space. And the resulting effect of that is it will make the atmosphere and the ocean as well more oxidizing. It will become more electron accepting relative to the inside of the planet. So what happens is the atmosphere and the ocean become electron accepting. The interior of the planet will become electron donating. That is a battery at the scale of a planet. And life is a way to release that stress that has been built up on the planetary scale. Yeah.
So,
of course, there are many details to still be discussed about this, but that's just an off-the-cuff statement to describing why life exists in a non-accidental way. It's just like when you have a planetary battery like that, it's not it becomes no longer an accident where something like life appears to relieve that stress built up.
well, I like what Mehrashad says here. He says neither chance nor necessity can generate information required for life's origin. So, like in your scenario, it seems like there could be a lot easier outlets for releasing this pent up energy than creating a genetic code that codes for all these proteins.
Yeah,
and
it doesn't explain the details of the energetic code. It's a whole other story as well. But I'm saying that like one of the, again, just to go over one specific scenario, like one of the most suspected aspects of life that is not accidental. Like we can like, for example, we can discuss that the fact that I exist can be described as a astronomical accident. There are a huge number of potential siblings that could have been born in my place. So my existence is like a contingent aspect of biology. But some of the things about biology doesn't seem to be an accident. Like for the fact that we have at the basic chemistry, what happens is that we see these what's called the citric acid cycle. The citric acid cycle involves a few chemicals, a few small carbon molecules that go into in a cycle. In us, it goes into one particular direction, which will spit out carbon dioxide, and that is why we exhale carbon dioxide from our lungs. But there are many bacteria that go in reverse. Carbon dioxide is sucked into the reverse citric acid cycle, and that builds up biomass. And one of the hypotheses is that cycle is not an accident. It's actually the path of least resistance to relieve the stress that I'm talking about. Like when you have a interior of the planet donating electrons and the exterior of the planet accepting electrons, what you have is what you end up with is a huge number of carbon dioxide on the exterior. The carbon dioxide on the exterior can accept electrons from an electron donor, like hydrogen, for example. But it's difficult for hydrogen to donate electrons to carbon dioxide in a very simple manner. Like when you put hydrogen, H2, and carbon dioxide together, they want, quote unquote, again, I'm using intentional language in a figurative sense, but please excuse my figurative language from now. Hydrogen and carbon dioxide, they want to react to become methane. That's actually an energy releasing reaction. When hydrogen donates electrons onto carbon dioxide, they want to become methane and release energy. But there is a huge barrier that prevents this. It's very difficult for hydrogen to react with carbon dioxide, but the way life does it, in the citric acid cycle specifically, or in the reverse citric acid cycle specifically, that represents a path of least resistance. It's like a lightning bolt, but in chemistry, where you have a channel for this stress to be released. So again, if you're interested in the things that I'm talking about, I highly recommend looking up a person called Eric Smith. But
to me, this is like very fanciful stories, and it's so
right.
Like I already have a story.
It's an off-the-cuff statement. It's not a scientific hypothesis. It's just an off-the-cuff statement that I want to relate to you. But you don't have to buy anything that I'm saying right here. No.
Right. But that's like, is there anything like if you like did experiments on these type of things and you were unable to get anywhere close to life, is there any point when you would accept the alternative that God created?
Oh, I think like if the alternative specifically says that God created something just by a miracle, like again, that goes back to my problem. Like if that's the hypothesis you propose, and there is no way to tell whether the hypothesis is factual or truthful or not, then we are stuck. We cannot we cannot come to a decision whether that scenario is truthful or not.
Right, but
But
do
if
you think you
if you
couldn't?
want to say
Yeah.
oh, sorry for interrupting. Go ahead. Sorry.
Well, I was just gonna say that the many features of life that show evidence of design and that you have a hard time talking about without using design language could be a clue toward that hypothesis.
No, I think the reason why we have a hard time talking about design or avoiding design terms, like you mentioned, recognition. I think it's just a quirk of language that we have inherited from my past. Like for example, in like we like in I don't know if you're familiar with quantum mechanics, but like in the on our scale that we experience, we can describe something as a particle or something as a wave. But when you go down into the quantum scale, we see things that are very unusual compared to ours. So we describe things in terms like wave or particle or both. But those are more analogies and metaphors rather than actual descriptions of what is going on. So we are really limited by the vocabulary that we have inherited from our culture, basically. That's what I will say about that.
Okay, Messerschmidt says bad news for Ness. New research shows early Earth had more oxidative atmosphere than expected.
That's actually a point in favor of mine, because I proposed that the Earth was a battery, but in the interior of the Earth, the rocky interior was electron donating. It is, or another word for electron donating is reducing, and the atmosphere was electron accepting. And another word for electron accepting is oxidizing. So yes, that's actually a point in my favor. The atmosphere was electron accepting, and the interior was electron donating or reducing versus oxidizing, and that is
Okay, and now he makes this point, even scientists with all their ingenuity can't create the simplest imaginable cell. Does that give you any, pause when you think that even with all of our intelligence, we cannot, even though we know the parts of a cell, we cannot even put a living cell and make it alive. Does that give you pause that maybe there's something, an intelligence at work that goes beyond?
But that in itself does not that in itself does not support any scenario over the other. It just says that the problem, like the whole point he's trying to make, of course, is that the whole problem of the origin of life is very huge. Like it's a very difficult problem that we still are trying to figure out. But the fact that we haven't figured it out as of now is not it should not be used to say like, oh, we give up. That's the whole point that I was trying to make earlier on, that we should not use our ignorance to make a conclusion from that ignorance.
But what if we used our intelligence? I don't think I'm using my ignorance to say God exists. I'm using my intelligence.
He's saying the fact that scientists are too ignorant to make a cell.
Right.
That is, it's not an argument that should be used to make a conclusion. It's just making the conclusion is that we are still have a lot of work to do to figure this out. That to me, that's the smartest way to approach a situation where you are incapable of explaining something. But even with all the progress that we have made in the how many centuries of scientific or thousands of years of scientific progress, we have not been able to figure this out, then we should. It's still not a good argument to make to say okay, this is the point where we give up and we no longer make any progress, and that's very bad in my opinion.
Well, again, I don't think it has to be limiting to science, but it could open your mind to a greater reality. So,
Well,
what I mean is, like, when I look around at the world, when I look at the orderly universe, when I look at a tree, when I look at a human being, when I look at a dog, I see God. I see a mind that created all these things. And that gives me a lot of purpose in life, knowing him, experiencing him. And so that's why I'm doing this, actually, because I want you to be able to see God.
well, I'd like, even though I myself am not a believer in any deity, but even though I don't think that sentiment that you just explained is incompatible with a naturalistic explanation.
I agree.
Yeah, it's not like even if tomorrow we finally figured out abiogenesis, we finally figured out how life came about on a non-living planet like Earth, like how according to even according to a believer in God, they can say like, oh, God has set up the universe with its in order such that scenario would have happened in a natural way still, like as it says in the Bible, let the earth bring forth life, compatible with that scripture.
Yeah, and I agree that it can be compatible. The reason that I do this, though, is because I do believe this origin story of evolution and abiogenesis, this is false. And I think there's lots of scientific evidence showing that it's false. So I don't need to make it compatible. And the problem is, I do think it's blocking people from having the best evidence that God has given us of his existence. You know, I've seen God do a lot of miracles, and I consider those nothing compared to my knowledge of God through the natural world. So
But even like you will still, like, even if we say, even if we found a natural explanation for the origin of life, like I don't see why that would stop anyone from saying like, oh, that the origin of life, even in a natural way, that is the miracle that God has orchestrated itself. It's not incompatible to say
I
that.
agree.
Yeah.
Yeah, I agree. I don't think it's incompatible. I just think
So
it's not true.
the only problem I have is like the moment when you propose a boom, a miracle happened and there is nothing else to discuss. Such scenarios don't help our understanding of what is going on.
Well,
Yeah.
I also think it doesn't help our understanding to insist on a natural process in spite of the scientific evidence. So what I think people are doing right now with the origin of life and evolution is they are ascribing supernatural processes to, like, they're giving supernatural power to natural processes. So it's like you're giving a power that it actually doesn't have. And so I think that is misleading.
Can you give one? I think I can also give one example of what you're actually saying. Like, I remember Francis Crick talking about, or maybe the other guy. I think
Watson.
Either Crick or Watson. Like, one of them said a remark about the origin of like the genetic code. Like, was a miracle that it happened this way, a frozen accident. And I don't agree with such a framing as well. So in that sense, I would agree with your statement on that. But in general, I don't think that scientists are like using supernatural explanations, and in such cases, when they do, I would agree that it's a problem when they rely on supernatural explanations for, because because those are not testable in as well. It goes back to my problem. I don't think an explanation where we cannot move further to have a discussion on would be helpful to our understanding of what is going on.
Okay, there's a lot of comments that have come in that I want to highlight. One of them, Wesley Coleman, I think he's asking you, what specific knowledge accumulation is going to stop under creation? Like, if creation is true, what knowledge accumulation is going to stop?
If we for example, if we assume that the origin of life, or anything else for that matter, or maybe let's say lightning, or it doesn't really matter exactly what we are talking about. Like, if you assume that it's just a miracle, or an unexplained something something happens that we cannot explain and we will never explain, then we can not move forward until somebody says, like, oh, here we can move forward. This is something we can test and build up upon. So, like, I'm not sure. Like, maybe if somebody shows a creation scenario that is testable, then we can have a close discussion on it.
Well, I mean, the thing is, like, what type of knowledge is important to get? Like, what I mean is, like, so is that going to stop technological advance?
No, like, I'm not saying that. Like, if we, for example, if we assume that abiogenesis was a miracle, I'm not saying that by itself will stop all of science, of course.
Right.
I'm just saying on that specific issue we are stuck on that issue. Yeah, alone. So maybe hopefully that will clear things up. I'm not saying that the whole of science will be dead if we assume one miracle. I'm not saying that, of course. Yeah.
Yeah. R. Brown said a while ago, there's no perfect evidence to prove evolution is true. There's only a boatload of great inductive arguments for it, but there are no good arguments for divine creation. Do you agree with that, Neslig?
Well, like, I'm not saying there are no good arguments for divine creation. I will say there are no sufficient arguments for divine creation. Like, there are many arguments, of course, that people will use for divine creation, like the William Lane Craig argument about the everything has a beginning, and everything has a beginning has a cause. Like, some of those arguments have been run around in philosophy, and some can argue it's a good argument, but I don't find those arguments convincing. So, yeah.
Okay,
so
what do you find?
If it's not like, if it's I'm not going to make a very strong argument saying that there are no good arguments for divine creation. I'm just saying there are no sufficient arguments to convince me for that conclusion. But yeah, that's my opinion on the matter.
Okay, and R. Brown, I'm complaining about this comment because I disagree that there's a boatload of great inductive arguments for evolution. I find most of the arguments for evolution entirely based on the presupposition of evolution. So I don't really see those great arguments. Now, Mehrashad said, neither chance nor necessity can generate information required for life's origin. Thank you. Casper said, well, who created God? God was not created. He always existed. And that actually...
sorry, can you show the other comment that you just showed?
Wait, this one?
I saw I briefly saw one.
This one?
That one, yeah.
Nick Lane is a great scientist. Is also good,
yeah. Yeah, you can. It's also a very, very good person to look up. If you're interested in the things I was talking about, he's a very good person to look up.
I'm not interested in abiogenesis because I think it's mythology, and I think it's a gigantic waste of time.
I'm not convinced in, like, well, a good example. I hope I'm not coming over as too condescending, but I'm also very interested in, like, Greek mythology and pagan mythology. Even though I don't believe in those things, I'm still very interested in figuring things out about how those belief systems operated and are still in operation today. So I just want to, even if you disagree with those things, I still want to, like, promote your curiosity, even if you disagree. I hope you will still, like, learn a few things about why people like Nick Lane are arguing these things.
Now I'm focused more on the evolution
Oh,
side of
yeah,
things because I feel like
fine.
I mean, like I'm never going to have the information that James Tour has, for example, and he's pretty much taken care of abiogenesis.
I don't think James Tour is very helpful in this. Like he tends to scream a lot and just
Why is
make
it always a complaint about? I mean, he screams when people don't listen, but the point is, why isn't anyone ever addressing his arguments? Why is it always something about, his beliefs or
well,
his method
the,
or whatever? Why isn't it ever about his science?
the problem is like when people like there is actually one example like I remember for a long time ago where James Tour showed a picture from a popular science article, and it shows a molecule, an organic molecule. And James Tour was in front of an audience saying like, "Oh, I don't know what this molecule even is. I don't even recognize what the hell this is, and it's sugar. It's a sugar molecule. Like, what are you doing? Like, to me, James Tour tends to like not carefully look at the things that he disagrees with. Like he jumps straight to the conclusion that it all must be nonsense, and he tends to overlook these very simple things to mistake a sugar molecule for something else, which he doesn't recognize. This was a very odd instance to see how even a very good scientist, like James Stewart is a very good scientist. He has the scientific background on organic chemistry, but to see him make such a mistake is very disconcerting.
Well, and I wasn't aware of that mistake, but I would say he may have made a mistake at one time or another, but he
Yeah.
has consistently presented major problems with abiogenesis, and nobody is willing to respond to them.
One reason why is like the rhetorical way he is arguing, like he goes around, like one good example is with the David Farina debate, and like I think you may have seen that too.
Oh,
That was
yes.
to me the whole thing was very horrifying as well. Oh, it's like I would argue that nobody who listened to that debate has come away to learn anything. Like even though David Farina and James Tour has shown papers on the screen and argued things, the way they were arguing and the way they were like constantly antagonizing each other, I don't see any productivity coming from it. Like, how does it help scientific progress in any sense? Like, it's.
I mean, if
That's my opinion on
anybody comes away thinking that, sorry,
Oh,
if
so go ahead.
anybody comes away thinking that James Tour was the problem in that debate, I don't know, something has gone wrong with the
I'm not
moral
saying
senses.
he was deep. I'm not saying he was the problem. I'm saying that him screaming and using just like rhetorical rhetorical arguments instead of calmly explaining the evidence or calmly explaining what he has any problems with it, and then that the whole discussion devolves into a screaming match. And of course, I'm also saying that the way David Farina behaved was also not far from optimal as well.
See, maybe we were watching a different debate, because the debate I saw was a scientist presenting scientific evidence and then just being insulted repeatedly. And then, even after being insulted repeatedly, Dr. Tour was still willing to say nice things about Dave Farina. And Dave Farina was only willing to continue to insult. That was, I mean, it was very
Well,
clear
Well,
what was
he was also
happening.
like he was also, like at one point he was like screaming at the at the like a whiteboard or maybe a blackboard. He was like, look over here, go, David. And then I was like, oh, well, how does it help when you're screaming like this? It's not a football match.
Okay, well,
Yeah.
we had a different
Um,
perspectives of that debate, but
yeah, I'm like again,
I
I
appreciate Dr.
even
Tour,
the
and I
yeah.
think he has
Mhm.
destroyed everyone on Origin of Life, and nobody can admit it, and nobody wants to admit the truth that origin of life is
I'm not.
yeah, it's an incredible, it's beyond a problem. It's really,
Well, I'm saying like destroying, quote unquote, someone in a debate is completely irrelevant to the scientific
no, I
process.
know. I'm not talking about, look, I'm not trying to, debates are mostly about
Right.
theatrics. Okay,
Exactly. That's exactly the
so
problem that I have. The whole debate was just theatrics, and
right,
I don't see how anyone like learned something productive from even though they showed some papers on it and showed some evidence. I don't. I think the main thing people got away from the debate is like, oh, David Farina destroyed James Stewart, or James Stewart destroyed David Farina. And like the whole thing was just a disaster, in my opinion. The whole thing.
yeah, I mean, and I think the mistake was that Dr. Tour did that debate, right? So that was where the mistake was. But it still stands right now that Dr. Tour has issued challenges and welcomed any discussions with, origin of life researchers, and no one is answering his scientific challenges to the origin of life. And even the one person, was it Lee Cronin, that agreed to meet with him and discuss it, and in a formal atmosphere, he didn't even address anything scientific. Lee Cronin completely ignored the science and just went on like a, theological thing. So nobody's willing to address the scientific arguments. But anyway, the point is, that's why I'm not really worried about the origin of life. I would never be able to even bring the knowledge of chemistry. I don't have the knowledge of chemistry like James Tour does. So he's already doing that. So I'm not really concerned about making that argument. But thank you for trying to stimulate my curiosity.
Okay, I'm glad to hear that. Like I'm not like, just to make sure, I'm not here to put you down on anything. Like I just want to like, again, as you to yourself, I want to promote your curiosity, and I also want to just make sure that you don't skip over important parts of a paper that you are wanting to critique. Like even if you at the end of your discussion of a paper, you still say like, oh, it's all nonsense, as long as you mention some of the proposed evidences in the paper, I would be very glad to see that, as long as you mention and discuss those things.
I will try to do that. I will try to think with your mind rather than mine, because I skipped over that, because to me it's not evidence at all. But I will try to see it with your lens of how you would see that as evidence.
I just like instead of adopting a mindset of like, oh, all of this is nonsense, just try to like, oh, so how are the scientists thinking about this problem? Like, oh, here is the scenario. What are they citing to in support, or maybe or to reject one scenario in favor of another scenario? And what are they talking about? Like, if they mention like, oh, in the paper that you mentioned, like they are talking about self-splicing group 2 interest. Oh, what are these things? Let me see what these things are. Oh, they are those. Oh, interesting. Like that's the process that you have to get yourself into, instead of beginning with saying like, oh, it's all nonsense, and then ending up in the same conclusion, it's all nonsense. So yeah, that's all I'm saying.
Okay, well, do you see any of the points that I brought forth today of how this might be a problem for evolution?
Oh, that's the spliceosome specifically,
Yes,
or
the alternative splicing. Is there it, or spliceosomes? Do you see, are you able to do what you're asking me to do by considering what I'm saying about the evidence?
if there's a paper out there that proposes a design scenario, like, or maybe several scenarios, and discussing some points of evidences in favor of one scenario or in favor of another scenario, I would be very interested into seeing it. But I'm not seeing any of those things from the design, intelligent design or creationism side. The only scenarios that I have seen comes from the evolution quote unquote side. They are the only ones who will propose a scenario, and then they just discuss in detail just the proposed reasons. Like, oh, this is the reason why we are in favor of this scenario, or this is one reason why we're in favor of another scenario. And sometimes there are, like in the case of the origin of eukaryotes, is also a problem. And there are many different scenarios proposed to explain how eukaryotes were originated, and there is still no solution to it. But at the very least, there are several scenarios proposed, but from the creation and intelligent design side, there are none, and that's the whole problem with the whole thing. Yeah.
But is there? Could it be true that okay, let's just say there's not a possibility of testing a certain creation scenario.
That's a huge problem.
But does that make evolution automatically true?
Oh, no.
Okay.
You're
So
completely right. If evolution,
I'm not making an effort to test creation. I'm trying to show you why evolution is not true. So is there anything in what I offered that could count to you as evidence against evolution being true, that might be working toward falsifying it?
Like, if you want to go to the very basics, like evolution, like you would have to say that like in order for evolution to work, you have to the inheritance has to be operating. But of course, you will accept microevolution, of course. So we can just skip the whole thing about microevolution. So, but if you look at the macroevolutionary scale of the whole history of the Earth, we would have to show that, like, one famous example of a test against evolution is a rabbit in the Cambrian. Something like that, or something similar like that, would be very difficult to explain under the current model of how life has evolved on the Earth. Like, that is very difficult to explain how a Cambrian can have been operated a Cambrian rabbit. Yeah, it's a very famous example of how you can test the evolutionary model of the history of life on the planet. Oh, sorry, go ahead. Sorry, I was rambling.
You know what's even stranger to me than if we found a Cambrian rabbit? The fact that we find all of this complex machinery at the very proposed beginning. So last week I gave you ribosomes. Ribosome in every form of life, even though it's incredibly complex, it's pushing the limit of like the age of the Earth in when that supposedly formed. Then today we have the alternative splicing and spliceosome right there at the beginning of eukaryotes suddenly appearing. So would you consider that as something that might be problematic for evolution?
I think you're making like a complexity argument. Like, so these things are too complex to have evolved. And I think like one of the most famous arguments, of formal arguments that have been made was by Michael Behe with this irreducible complexity, right? So are you making such an argument that these things are irreducibly complex to have evolved?
Not just irreducible complexity, that is part of it, but additionally, the sudden appearance of it. So that's problematic, the sudden appearance, right? So because evolution needs gradual evolution. Well, it from the beginning, it said with ribosomes, it's from the very beginning, the ribosome is said to have existed.
Well, in that case, I think they are saying, like, in the last universal common ancestor of all cellular life, I had a ribosome. But we are not. But I don't think they are saying, like, at the moment of when the planet formed, we had.
Well, it's getting close to that because the life has moved back. The origin of life has moved back to like 4.1 billion years. So we're getting close there to the beginning of Earth.
That still leaves like hundreds of millions of years before the formation of the planet. So it's
okay.
I think the one issue that we have is we are to mind the scale that we are dealing with. Like, to a geologist, 10 million years is nothing to a geologist, but 10 million years, that is a long time for biology. So we have to be mindful of how we are describing the scale of the time that we are talking about. Yeah.
Okay. Well, Nestlek, it's been so nice talking to you.
All
There's
right.
somebody
So,
coming
sorry for the
backstage right
long.
now. No, I
All right.
loved it. This has been
Mhm.
wonderful. This is exactly the type of conversation that I want to have. So thank you for making my day.
Oh, but glad
And
to hear that.
let's pick it back up if you spend time. I'll read that paper that you mentioned,
Oh,
and
yeah.
if you read it, then we can talk about it. And I'm
Yeah,
gonna
I will
let
make it.
Big
I
Back
will make
Mama
it.
on.
Like, again, I apologize if you on the Peaceful Science Forum, anybody can join, and of course anybody can be very, very critical of each other. So, and sometimes it can be off-putting. So, but if you don't want to deal with anybody else on the forum, just ignore them and just state, I don't want to interact with you. I just want to interact with one person or those people. You are free to do that. So, of course, but. Or if you don't want to be on the forum, it's also up to you. I will just make a post on the forum to make a few notes about the alternative splicing. Is it okay with you?
Yeah,
All right.
that's great. And I'll read it. Yeah, for sure.
All right.
Okay. And here was one comment that I think was intended for you. What about the 40 animal phyla appearing suddenly without any obvious
Well,
ancestry? Good
the
point.
problem is, okay, can I answer that question really quick,
Sure.
or do you want to move on to the next person?
You can answer it. I brought it up
All
for
right.
you. So
So the fact is that most animal phyla don't have a fossil record, so we can't really tell when exactly those begin. But we do have many do appear in the Cambrian, and a few do appear before the Cambrian as well. So that is also a point of to make about that. But the more important point is that phyla is also a very arbitrary categorization as well. So like when you talk about a phyla today, for example, our phyla is named chordates, and that refers to a structure in the spine of our backbone, for example. That's a chordate. The animals they include in chordata are mammals like us. They also include reptiles, amphibians, fish, birds, and as well, sharks and such. Those are all included in the like anything with the backbone are included within the phylum Chordata. But when you go back to the Cambrian, the phyla looks very different. All we see in the Cambrian are like these tiny little fish-like creatures. You can barely call them fish. Like most fish today have a backbone, but during the early Cambrian, there are no backbones even yet. They are like if you want to see something similar, there is a creature called picai. No, not picai. The lancelet. Are you familiar with the lancelet, Rebecca?
no.
I can maybe put a link to a Wikipedia page about lancelet. Let's see here. Also called Amphioxus. Let's see here. I will put it in the live chat.
Okay.
I
It may not let you post a link, but I should let the other
I'll put
person
the
come on.
name. The name.
Yeah.
That's the name of the creature. It's a lance. It's also included in our phyla, and basically the only chordates we see in the Cambrian, especially the early phases of the Cambrian, we only see things like those. We don't see the fish that you are familiar with, like things like sharks. They appear after the Cambrian has even ended. So yeah. So we need to be like when we say when we say a phylum appeared in the Cambrian, we are not saying that all the members like frogs, fish or mammals, they have not appeared in the Cambrian. Yeah.
Right, but that there's lots of, and I do want to let the other person on, so I don't want to spend too long on this.
Sorry for
but I
the
do
rambling.
want to say real quick, but we don't have any examples of the vertebral column developing. So, like, how did we get this vertebral column? We have no ancestors, like predecessors of those early animals, like even trilobites. We don't even have a single like transition to a trilobite. So there's a lot of problems with the fossil record that we could talk about.
I do have some disagreements about those, but I don't want to put the person on hold for a long
All
time because
right.
I've been a long time.
We'll
But we can talk about those things at a
we'll
different
do it
point. So
again.
just yeah. All right.
Okay. Thank you so much. Have a good day.
Bye bye.
Bye. Hey, big bad mama.
Hi,
Glad to see
Rebecca. Am I
you
coming
here.
through? Good to see you.
You too.
Am I
Do you
coming
have
through okay?
evidence that you want to share for evolution?
Yeah, I'd like to talk about biogeography, which is the study of the distribution of species and ecosystems in geographic space and through geological time, and specifically the marsupials.
Awesome.
Why the
Okay.
marsupials are limited to Australia mainly, and we do have marsupials in North and South America, including a lot of fossilized species in South America. Go ahead.
No, that's awesome topic. And I just want to put up a couple of comments, just zooming through them really quick because
Sure.
I like these. Mavis said, look at the trees. Yes, that is my argument. Look at the trees. Mehrashad said, if all intelligence is incapable of making a cell, how do we expect a mindless process to do it? Exactly. By the way, Mehrashad, this would be great if you had a channel in Persian, since it seems like this is a topic that you're interested in. Tom said, knowing God created the universe didn't stop Newton. Thank you. And Casper says, I agree with Rebecca. Looking at a tree can fill you with awe. Nature and its complexity is very exciting indeed. Okay, well, I already put that one up. And Tom said the chemicals that make up life are unstable. They break apart with time. You're trying to say the chemicals come together by accident over time when the opposite happens. Thank you, Tom. And I'm sorry I didn't put that up when Nestlig was on, but appreciate your comments. Okay, go for it, Big Bad Mama. We're doing biogeography. Great.
Yeah, so we're talking about Australia and the marsupials, specifically why Australia just has marsupials mainly. And, the DNA evidence has shown they've done studies that support an American origin for marsupials. And it's explained through the breakup of Gondwanaland. There was a single Gondwana migration of marsupials from South America across Antarctica to Australia. And we know when that breakup occurred, 35 million. They migrated about 50 million years ago, and they broke apart. These continents broke apart, isolated Australia around 35 million years ago, I believe. So that breakup explains specifically why you just have marsupials in Australia. And also, they predicted that they should find marsupial fossils in Antarctica since they were connected. South America, Australia, and Antarctica were connected. And indeed, they did find fossils, marsupial fossils in Antarctica. And this is when Antarctica was, it wasn't at the South Pole. Do you understand? It was tectonic activity had it further north. And so it wasn't a polar climate back then. But do you have anything so far about that?
How is this evidence for evolution?
Well, it's evidence because the marsupials evolved specifically on Australia, and all the proliferation of marsupials in Australia were from a single migration of a marsupial organism. So not only do they have the DNA evidence that they've collected from modern marsupials and compared it with South American marsupials, but you have the fossil record in all those locations. And, Kent Hovind claimed that humans brought the marsupials to Australia from Noah's Ark, from the Near East and stuff, which, what was their food sources? What did the koala bears eat? And that kind of thing. So it is strong evidence for evolution of that entire type of mammal.
Okay. I'm trying to figure out. This sounds like it's mostly an argument against young Earth creation rather than an argument.
Well, it's that,
for
of
evolution.
course, but
Okay.
no, I mean,
So I'm not really interested. Like I am a young Earth
no,
creationist, but I'm not
but
arguing for
you have to
young Earth
understand
creation.
all the marsupial forms that came that exist in Australia evolved from a single migration of a species of marsupial. So you would say that the evolution of marsupials in Australia into all their present forms was evolution, massive speciation event.
Okay, well, see, I actually think marsupials are a huge problem for evolution. So, I would need to understand why this would be evidence for evolution when I see it as evidence against evolution.
Why
And
do you think it's
I'm right
evidence
now trying to
against
look
evolution?
for a
Okay,
picture that I'm trying to look for a picture
let
in
me,
this
can I
book.
add one more thing,
Yeah, sure.
Rebecca? So, you know, if you were to take, and I know you might not like this discussion, but if you were to take a global flood event, you would have those marsupials making their way from the Near East to Australia and migrating that way. But what we have in Australia is we have several fossil events, several fossil locations that show that, with ages that show the development of the marsupials in Australia, the evolutionary development. So you wouldn't have those layers that have the fossil marsupials and also have, um, why would you have those fossilized sites of marsupials there before the flood, and then all of a sudden the marsupials came back after the flood? Do you understand? How,
Yeah, but I mean,
that's a
there's
big problem.
marsupials
That's a
found
huge
in
problem.
there's marsupial fossils found in South America and North
Right,
America, various
because of the
places. So,
Gondwana
yeah.
because of Gondwana land. Yeah, because we were connected with all those land masses were connected. So this ties into plate tectonics and evolution. It's very powerful evidence, biogeography.
Okay,
And maybe you
well,
want to look
again,
more
it
into it and we
sounds
can talk
more
about it
like
more
you're
later. Um,
well, that one
Go ahead.
sounds more like an argument against young earth creation. So, um because I think let me share what to me is
Sure.
problematic. And I love this argument about marsupials as like when people bring up marsupials because it is such a problem for evolution. So I don't see how whatever you could offer with the like geographical distribution is going to solve this problem. So trying to show this right here. So we have at the top is a placental mole, picture of a placental mole, and then you've got a marsupial mole, right? Now,
Right.
those look very similar. And if you had to say which of these three, like if I show you these three organisms, one being a humpback whale, and then the placental mole and the marsupial
I
mole,
understand. Yeah.
which ones look like they're more related?
Well,
The
of
moles,
course,
right? If
if
you
you're
had to
just
group
going
them
by
together.
looks, yeah, it's the moles.
Right,
But if
exactly.
you were to
Right.
study if you
So
were to study their physiology, you wouldn't make that conclusion because the
yeah,
whales
and
and the placental mole raise their young exactly the same way and give birth exactly the same way. So yet you need to
well,
go into it more than the detail of just the looks of it.
I know, and but see, to me, this is a major problem with evolution is that you've got
It's really
these
not.
two animals being more closely related, and yet
Those
somehow this evolved on a totally different tree, and it evolved to look exactly the same
Well, they
as the
evolved to burrow
placental
in the
mole.
ground, right? They're moles, so they burrowed in the ground, and so they occupied similar niches. So you had all the Australian marsupials fill niches that elsewhere would be filled by placental mammals. So anything that's burrowing in the ground to make its living eating insects, insectivores, or what have you, whether it's a marsupial or a placental mammal, is going to look alike because it has to burrow in the ground. It's going to have small eyes. It's in the dark. It doesn't need like a lot of fur and that kind of thing. So that's why they've developed to look similar to each other, but they had completely separate evolutionary paths. That's my answer to
Right.
that.
And so the reason that's problematic is because you said evolved to. And so it sounds like there was a goal in mind. They wanted to burrow in the ground And so they evolved to burrow in the ground. And when we're talking about evolution, we're trying to get random mutations to somehow coordinate, thousands of mutations to coordinate together to get a feature that would be
but
worthy of be selected, right?
You're missing
that
a big. Yeah, I understand. I'm sorry I
Okay.
interrupted you.
So this is problematic because we have this over and over again. Here's the placental mouse, here's the marsupial mouse, and yet we have to conclude, even though these two look exactly alike, we have to conclude that these two are more genetically related.
Okay, but which ones are more alike if you actually study their anatomy?
And that's the problem, is that
Well, I'm asking you. I'm asking you that question of if you study the two moles and the whale, and you do a study of their organs and sexual organs and that kind of thing, which ones are you going to come to the conclusion are more closely related when you do that?
Well, except for their reproductive methodology, their reproductive
Which is a major
method.
thing. I mean,
Right, but are you really going to say that the anatomy of a humpback whale is closer to the anatomy of a placental mole than a placental mole and a marsupial mole?
yeah, I would say the way they the way they give birth, the way they raise their young, which are major features in evolution, are more similar in the placental mole and the whale than in the marsupial mole and the whale.
And that's why evolution has brought people to nonsense conclusions. And this is not a problem. And it is true, absolute nonsense, because you guys are trying to group animals together
okay.
by similarities and say they developed in such and such a way. But the problem is your trees don't work. They don't work morphologically. The genetic with the morphological don't work. The protein with all the trees contradict each other. There's no nice, neat tree of development.
Do you mean that? Well, one thing that you said was this was just based on random mutations and accumulation of random mutations, but you did leave out natural selection and the role that the environment plays in favoring certain mutations, and that isn't a random thing. That's
Right.
a very directed thing. And I always hear this from creationists that, oh, you guys just believe everything was created at random. And anybody that's honestly talking about evolution, and I'm not saying you're not, but has to mention what the environment does in evolution. But what is your answer to how the marsupials got to Australia?
I don't have an answer, and I will say that is something that's very curious to me. I think it's problematic for my belief. So
it's very problematic.
Yeah, it's problematic for young Earth creation specifically.
Well,
right?
and for believing in the literal global flood, it's very problematic for that, because you have to explain how a pair of, how many basal kinds of marsupials were on the ark and how they got from Mount Ararat to Australia. And was it one pair of marsupials that made it and then evolved into all the species that exist today, or were there multiple pairs of marsupials that made that journey? And how could they have made that journey, without getting preyed on? What were their food sources? Eucalyptus and different things that are very specific food sources to Australia. But I'll leave you with that.
No, but that's the thing is, that is, like I said, that is the story I believe. And I recognize that there are some problems with it. I'm not presenting to anyone that as the scientific view of how we came about, but evolution is being presented as the scientific view of how we came about, even though it has many, many contradictions. And marsupials is one of them, because just those two examples, I could give you many more examples of
which I explained.
Marsupials.
I explained.
No, you did not explain
this.
Yes,
I did, because it's all about niches. It's all about filling niches. And
right, but let me have a chance to respond to what you
Yeah,
said.
go ahead.
Okay. Because we went directly into the young earth stuff.
Sure.
But I have a counter to your thing, because you're saying, well, it's not just random mutation, it's also natural selection. Yes, but nature can only select from what is already present. So what you're saying is that by random mutations, we were able to get moles that look just alike, mice that look just alike, big cats that look just alike, all kinds of creatures that look just alike. And if you didn't know they were placental versus marsupial, I mean, there's even like a
Well, why wouldn't you know
marsupial
that?
saber-toothed tiger, right?
True.
So
Yeah, true.
like, this doesn't really,
But they don't look, a marsupial saber-tooth tiger does not look like a saber-tooth tiger from North America, before the Ice Age or what have you. There are big differences in those skeletons and what they look like. So,
and that may be true for those, but there's numerous examples of
okay, but
marsupials that are
let me just
exactly
say,
look like the placenta.
scientists study more than just the looks of an organism. They're studying their anatomy, they're studying their reproductive systems, they're studying where they live and all sorts of things. So your example is just going off of something looks like another thing, which doesn't look like this other thing. And scientists delve quite a bit deeper than just saying this looks like this, looks like this. It's what's inside the organism, as opposed to just what's on the exterior.
Right. And what I'm saying is that diving deeper, the diving deeper is fine. But the problem is the presupposition of evolution causes people to come to ridiculous conclusions. So the idea, okay, so I mean, show a 2-year-old like those pictures. Which one is more similar?
We're not, a 2-year-old doesn't understanding the science. Science is
A
complex.
2-year-old hasn't been brainwashed into this evolutionary story.
Well, you could say the same thing about religion. I mean, let's face it. I mean, kids are brainwashed on religion far earlier than they are on evolution. All that, since you went there.
Yeah, I mean, actually, I mean, if they are in a religious household, maybe,
Right.
But if they're going into school at age 4, then they're usually getting brainwashed at that time.
Yeah, nobody learns evolution at the age of 4, Oh, here's where you match the circles and the squares. By the way, Darwin said, nobody did that when
No, they start
4.
with the dinosaurs being millions of years old, and
Right. Well, that's another topic. And
I
maybe
know that I was
indoctrinated
by 5th grade. I can tell you, I was deeply indoctrinated by 5th grade.
Poor thing.
Yes.
Well, thank you, Rebecca. I enjoyed talking to you, and I'd like to do this again sometime.
Okay. Thank you so much. Appreciate you coming on. Okay.
Take care. It was fun.
Care.
Thank you.
Thank you. All right. Well, thank you all. I think we've been going for 2.5 hours, so this might be a good time to stop. Appreciate everybody who came on, and yeah, next time bring your evidence for evolution, and I'll bring my evidence against evolution. So again, alternative splicing. I think that is a big problem for evolution. I gave my reasons. I would love to see Nestle come back on that, and talk to you guys later. Bye.